Everything you need to know for GCSE Biology Paper 1 Part 3, make sure you go watch the other parts. Starting off with digestion, and the mouth is where it all starts, because the mouth is where the food is ingested and it's broken down into smaller pieces, increasing the surface area for digestion. The salivary glands secrete saliva, which contain enzymes like amylase, which break down starch into smaller sugars like maltose. Then we have the esophagus, which is the tube which connects the mouth to the stomach, and this tube, the esophagus, uses peristalsis, which is a contraction of the muscles that make up the esophagus, which push the food down from the mouth all the way down to the stomach. The walls of the stomach contract and they churn up the food, continuing that mechanical digestion that we saw in the mouth. And the stomach contains hydrochloric acid, which 1, kills any pathogens inside the food, and 2, it provides a suitable pH for the stomach enzymes like protease to work. This over here is the liver, and the liver produces bile, which is then stored in the gallbladder, and bile is super important for 1, neutralizing stomach acid as the food leaves the stomach and enters the small intestines, and 2, bile helps to emulsify fats, increasing their surface area for lipase enzymes to break those fats down. The pancreas is also your best pal for digestion because it produces protease, amylase, and lipase enzymes to break down proteins, starch, and lipids in your small intestines. In the small intestine, that's where your digestion is complete because of those enzymes breaking down those food molecules, and the food molecules and water begin to be absorbed into the blood in the small intestines via the villi. Then the food moves into the large intestines, which is where the remaining water is absorbed out of the food, and the food moves down into the rectum and the anus. Then poo. We should remember that enzymes work via a lock and key hypothesis. So a substrate must have a complementary size and shape to the active site of the enzyme in order for it to fit, to form an enzyme substrate complex, and therefore to break down this substrate. And we need to know about the factors affecting enzyme function, pH being one of them, and every enzyme has an optimum pH. We spoke about the protease enzymes inside the stomach, where the hydrochloric acid brings the pH down, making it optimum conditions for the protease enzymes to work. Every enzyme will have an optimum pH level at which they work. Anything below that will mean it's too acidic and the enzymes denature. The active site changes shape and the substrate no longer fits in. And any pH too high, too alkaline, the active site denatures, substrate can't fit in, and the enzyme doesn't work. Temperature is another factor that affects enzyme function. If the temperature is too low, there's not very much kinetic energy. So the likelihood of a substrate randomly hitting an active site is very low because they're moving around so sluggishly. But as the temperature increases up to the optimum temperature, the kinetic energy of the substrate and the enzymes increases, so the likelihood of a random connection of a substrate and an active site increases. However, as the temperature gets too high, the activity of the enzyme decreases again, because if the temperature is too high, this denatures the enzyme, changing the shape of the active site, meaning that it's no longer complementary to the substrate. And finally, as substrate concentration increases, the rate of reaction also increases because there's more substrate, therefore there's a greater likelihood that a substrate will collide into an active site. However, this is only up to a certain concentration of substrate, after which it levels out and there is no more effect because all of the active sites will be filled, so adding more substrate doesn't make a difference. It's like if I had 3 Christmas cards to write and I had 2 friends help me, so all 3 of us are writing a Christmas card each, that will speed me up. But if I didn't get another 30 friends, it won't make any difference to the speed because there's only 3 Christmas cards to write. And you must remember that starch is broken down by amylase into smaller sugar molecules, that proteins are broken down by protease into amino acids, and that lipids are broken down by lipase into glycerol and 3 fatty acids. And for food tests, I want you to remember that the test for starch is iodine, and iodine goes orange-brown to blue-black in the presence of starch. Benedict's is the test for sugar, and it goes from Benedict's blue to a traffic light system of green to red, red meaning lots of sugar present. Ethanol is the test for lipids. It goes from colorless to a cloudy emulsion in the presence of lipids. And burette test is a test for proteins, and it goes from burette blue to lilac purple in the presence of proteins. So all of the reagents that start with B start off as blue. Burette, Benedict's, both start with B, both start as blue. Remember that. And that's the end of part 3. Make sure you guys drop a follow if you haven't already. Send this to a friend so that they can also revise this and watch the other parts if you haven't already.